Welcome to another episode of the DNA of Things, where we explore the intersection of science, technology, and health. I'm your host, Dr. Jeremy Koenig. Today, we have an incredible guest, Dr. Urban A. Kiernan, the visionary founder, chairman, and CEO of iMetabolic Biopharma. Dr. Kiernan has a deep background in bioanalytical chemistry, proteomics, and drives innovation in cardiovascular health. He has spent decades pioneering new ways to fight disease through the development of therapeutics. We dive now into his remarkable journey from the early days of proteomics to building cutting-edge AI drug discovery platforms. Now, in this episode, we explore how Dr. Kiernan's path took a pivotal turn from academia to industry and through personal tragedy. That's the backdrop of a story around the science behind proteomics and its future for health and human performance. His passion is emergent in this conversation as so far as transforming drug development and the future of therapeutics for all. We discuss the importance of personalized healthcare, how advances in bioinformatics are revolutionizing treatment, and why understanding lipid metabolism holds a key to treating diseases like cardiovascular issues, diabetes, and even Alzheimer's. So whether you're a healthcare professional, a biotech enthusiast, a tech bio enthusiast, I should say, or simply someone who cares about your health, this episode is packed with knowledge and insight that could truly change your life. Let us dive in. Dr. Urban Kiernan, the founder, chairman, and CEO of iMetabolic Biopharma Corporation, where he drives innovation in the biopharmaceutical industry, focused on cardiovascular health, strong background in bioanalytical chemistry, passion for improving patient outcomes. Dr. Kiernan is also the host of the Decoding Health podcast, where he explores the intersection of science, healthcare, and the business of pharmaceuticals. Welcome to the show, Dr. Kiernan.
Thank you very much, Dr. Koenig. Really appreciate the opportunity to work with you again and being able to go and basically be on this side of the microphone and be interviewed. So it's a little tit for tat type thing, but this should be a lot of fun.
Yeah. I reflect back to our last conversation, and as you call it, being on the other side, I was like, wow, that's quite different. It's easy to kind of sit here and look smart when you're asking all the questions, right? So my turn to be smart, but that won't be too hard because we've had that great conversation in the past, and you've done some pretty stunning things. One of the things yes, of course, my pleasure. One of the things we're doing here at the DNA of Things is building a community of conscious creators. And you've probably seen, we've had some really incredible guests on the show already. And there's so many, as I say, just kind of natural connections between the network. And so it really pleases me to bring you on. I don't think I've had anybody from the proteomics background. So really, really keen to sharpen my brain around that topic. But before we dive into perhaps all the techie stuff and the innovative projects that you got going on now, always keen to know about the human behind the story. So your journey to biotech, how far back should we go? When did you get essentially called to your current mission? And what were some of the challenges along the way that led to your overcoming and being where you are and who you are in the realm of biotechnology and life?
Well, how much time do we actually have for this episode? No, that's a great question. And I really love sharing my story for multiple different reasons. And I'll kind of cover those as I just kind of talk about this stuff. So I'm going to just turn the clock back to like 1998. It's a different millennium, but nonetheless, started graduate school. And, a bright-eyed, bushy-tailed, going into graduate school. And the reason why I start the story there is because from a career perspective, it's all about opportunity. And, for the listeners out there, you are provided opportunity every single day. It's just whether or not you have the wherewithal to recognize them and then the courage to be able to execute on that opportunity. And that is very much my story. So, started here at Arizona State University, made that home, getting into a technology called MALDI, matrix assisted laser desorption ionization, time of flight mass spectrometry. And so this was an approach that was becoming very, very innovative at that point in time, because it was large molecule mass spec. Up until this point in time, everything was like small molecule, and there's unique aspects of the biophysics or the physics, the ion physics regarding that just it up until the development of electrospray ionization and then MALDI, just large molecules were impossible to do. And that opened up an entirely new frontier of the way you could go and interrogate and analyze proteins. And so this is also at the tail end of the Human Genome Project. And, that's absolutely relevant because the lab that I went into was developing mass spec technologies for analyzing DNA. Actually, it ended up on the cover of Science with some of my predecessors in the laboratory. But it also ended up because there was already so much investment going into that space, and there were already so many people contributing to it. You could already see that this is already getting saturated. So how do you turn around and differentiate yourself? So rather than going and focusing on the actual genes, let's turn around and focus on gene products. And so if any of the listeners out there are unfamiliar with proteomics, you're familiar with genomics. And so you're analyzing the entire genetic makeup of an organism in theory. But what do genes actually do? And it's really simple. They encode for a protein. So a protein is the functional form of the gene, whether that function be structural, transport, signaling. The sky's the limit regarding what these biomolecules actually do. And this was a new frontier, to be honest with you. And so there was a lot of lessons that were being learned in the infancy of proteomics. And again, back to the whole storyline of opportunity, I was blessed in which I was given an opportunity to start working in industry while I was in graduate school. And the majority of the work that I did in industry actually counted towards my PhD thesis. And so the group that I was with at a company called Intrinsic Biopros over here in Tempe, Arizona, it was a different approach on things, because just like in the mindset of genomics, again, in theory, looking at all of the genes within a given organism, the initial goal of proteomics is to go and look at all of the proteins that are expressed in an organism at a given moment. And when they solved the initial run in the Human Genome Project, one of the interesting things that came out of that was actually how simplistic the human genome actually was in that initial round. And, there was a lot of controversy around that, because there's genomes of other organisms, and there were some worms that had more complex genomes than human beings. And it's like, this doesn't really match up. And so this flipped over into this, well, the answer has to be in the proteins. And it led to this entire slew of people trying to go and attack the protein analytics, the proteomics aspect, in the exact same fashion that they were tackling the genomic aspect. And that led to this thing called non-hypothesis based, I can't even say without laughing, non-hypothesis based science. And basically what it was is just people were generating gobs and gobs of data, like terabytes full of data per experiment, with the intent of trying to find some sort of answer to something at some point in time from that data. And that's just a very, very poor way of setting up an experiment, in my mind. And so the way at that group at Intrinsic Bioprobes, or IBI, it was a targeted approach. So we were the pioneers when it came to immunoaffinity mass spectrometry. And so we were actually the first ones to go and take antibodies, put it onto a solid support. And these were in the forms of these affinity pipette tips. We can interrogate a biological fluid like human urine or plasma, go in, pull out a particular protein of interest, elute it, clean it up, elute it from that capture device. So the way the antibodies work, again, for the naive listener that's out there, basically acts like a magnet, but it's very, very specific. So that antibody on this device is only going to pull out one specific protein from all of the thousands, millions of different possibilities that are in the human that are in the human proteome, and be able to go and put that into the mass spectrometer to go and look at what that protein and all of those proteoforms of that particular protein actually looks like in a single analysis. Pardon me there. And that was really, really interesting because, when, you go in, excuse me 1 second. You go in, you get your blood work done. Everybody does it. When they run several, proteins as a part of that, you get a number back, and you just associate one protein with one number. But herein lies the rub. Each protein resides in in the human body in multiple different forms. And that is, you can't get that level of fidelity. You can't get that level of detail when all you're doing is just quantitating that particular, the totality of that particular protein. And so there's a tremendous amount of information that we were able to dissect, excuse me, out of those proteoforms, and actually correlate those back to disease states. And, that led to a number of, publications, patents. It was really fun starting off my career that way, because it literally felt like you were going to the moon every single day, because you were seeing proteins that nobody else had ever seen before, like C-reactive protein. I was the first person on the planet to see that in a mass spectrometer, CKMB, and again, all of these things. There are so many different variants of these proteins, and some of them are because of natural degradation products that take place, because you have enzymes floating around in your bloodstream. And this is one of those things with, everybody's on the semaglutide, the Ozempic craze, and, they call them GLP-1s. Well, they're not really GLP-1, they're GLP-1 receptor agonists. It's because GLP-1 as a therapeutic is just crappy. It's because they are digested very quickly in the bloodstream by these enzymes. And so, that was some of the initial work that I did in that space many moon ago. This is like 2002, before there was any of those approved drugs. I'll never get credit for that work that we did, but those are some of the things that we were experiencing. Is you've got this active biological fluid, and your body is producing these signal proteins, and your body is just somewhat summarily degrading them, giving them very, very short lives, and they can't even turn around and do their job. And so you need to do, in the biopharmaceutical space, a lot of, trickery in order to be able to circumvent those natural biological processes. And so, had a really great career doing all of that, got funded by the NIH, big grant from the NIDDK, that's the diabetes section of the National Institutes of Health, and really got to work with some strong key opinion leaders and learn a lot about lipid metabolism as a part of that. Because at that point in time, in the study of pre and type 2 diabetes, there were two schools of thoughts. There was the inflammation group, and there was the lipid metabolism group. And there were way more people studying inflammation. I think it's just because there are more ways of studying inflammation than there was in lipid metabolism. And that was actually very fortuitous in terms of where I am at in my career, because that gave me a huge learning curve, and being able to go and do the types of very targeted experiments that I was doing, and doing it for very, very specific purpose, going into disease states. And so it's really interesting talking about these proteoforms and looking at some of these these fat associating proteins and being able to see things like oxidation. You see these 16 Dalton shifts because that's the weight of oxygen, and you're able to go and see the level of oxidation of a given protein and seeing how these subtle changes that if you did a conventional assay and just got a total number, you would never see this, and how those things correlate with specific diseases. Now, that was, as I said before, was a very, very good beginning to my career. But things and very big into like diagnostics, and let's go and diagnose disease early and, set that as the foundation for, new therapeutics and treatments and everything. But there's a personal aspect to this. It was back in 2006, my wife and mother of my two daughters passed away from cancer. And that was a very, very humbling experience because, so far in that entire career, you're on this trajectory in terms of doing some really, really awesome stuff. And then you come to realize that everything that you were doing was for actually nothing. In reality, it's like biomarkers are great. Diagnostic molecules are great. But unless they give you a point on a decision tree to go and either intervene route A or B, then it's a wasted point. And so that was actually the point in time where it kind of like really smacked me in my face regarding I really need to understand the therapeutic aspect of things, because that's what is going to turn around and drive change. And so, long story short, IBI, the company I was with, we were acquired by Thermo Fisher Scientific. Jumped over to Thermo Fisher Scientific, did the global biz dev thing over there for several years. And then, long story short, left that position because of my situation. Remember, single dad, two daughters, traveling the world, doing this job. Things were just kind of compounding on me and I needed to take a break. So I left in 2016, turned around, really focused on redeveloping me, focus on health, wellness, things that I could personally control and take back in my life, because it's really easy from the hustle and bustle of trying to keep up with the corporate world to be able to lose yourself in what is the prioritization around health and stuff. And so 2017, that allowed me to found iMetabolic Biopharma, or IMBP for short. And we're off to the races in terms of innovating novel therapeutics in basically, I'm just going to say, all things lipid metabolism based, with a heavy emphasis right now on cardiometabolic, cardiovascular, and neurodegenerative diseases, because there is this underlying theme when it comes to a metabolic aspect that is fundamentally missing in the way they approach the disease. So, in some ways, my trajectory in terms of my career, exposure to science, it's the same story as a lot of other people, but then there are those little unique caveats that just give you that little twist that you would never have expected. Again, all beginning with, wow, getting a job while in graduate school and being able to be on the forefront of that innovation and learning about patents very early in my career, getting those publications, writing my own SBIRs, that's some, life lessons as a scientist that, you can't put a price tag on that stuff in terms of learning all of those things and then being able to go and just translate, and then even to the acquisition standpoint, being a part of a Fortune 500 company and seeing how businesses run, being able to interact with the biggest biopharmaceutical companies on the planet, and basically learning, what things are done right, what things are done wrong, and how do you turn around and improve upon these processes, and then being able to integrate that as a part of what is part of the core essence of INBP, which is efficiency. It's a lot of drug companies. It is a very, very expensive proposition to go and take innovation and taking it to an FDA approved drug, but it doesn't have to be as expensive as it is if you work on efficiency, remove redundancy, and just have a very, very good clear cut plan and mitigate the oops moments of critical mistakes that you should have known better to not have do in the first place.
Mm-hmm. Wow, what a journey! I'm interested to know, just kind of a few more questions on the introduction. Like, why science? Why did you go into science specifically?
Oh, geez. So my mom was a nurse, so that's like the health aspect of everything. And my mom's side of the family, there's like a bunch of, physicians and everything. So
Mm.
that, and I'm half Filipino. So if anybody watches like Joe Coy comedy and stuff and like what children have to sacrifice to appease their parents, that's a very, very real thing in the Filipino culture. So I'm just going to kind of throw that out there. And then my dad had a master's degree in chemistry. So it's one of those things that I just kind of like grew up with. And I
Yeah.
remember vividly as a little kid, my dad taking me into the high school chemistry lab and we were making things blow up.
Yeah. 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 So I'm always fascinated at first there's like, our education system, we tend to specialize, I think, young. I mean, at least that's the case in, Canada. I mean, although you have a comprehensive education and in both public and private school, there is kind of this push one way or another. For me personally, it was, I think it was the Mulroney government at the time, kind of sent around, what would you call them, representatives, to basically say that really math and science is the only way to be successful. And I personally loved writing. I love creative writing, and I think that the creative process in general can be applied to the sciences. And I think about, of course, I chose that path, but I listened to you speak to, some of the parallel things that I experienced on the side of genomics, you experiencing them on, proteomics. Well, again, most notably the completion of the Human Genome Project, this statement that, well, it's all junk or mostly junk because, most of it doesn't code for proteins. And then, as you say, there's this, I like to use the phrase, what is it called? Hypothesis outcome based science, which is the same thing as what you were saying is non-hypothesis based science. But it is getting the landscape of things that, we don't know. In my realm, it was, I did a lot of work in microbial diversity, and there was what was called the uncultivatable majority. So you can go out with the advancement of the improvement, let's say the DNA extraction protocols, you could get DNA extractions from the most, extreme places. So mine tailing sites or hypothermic vents, Great Barrier Reef, and you don't need to grow the organisms. You can just sequence the DNA, and it is, as you say, it's this big kind of stamp collecting project, and you're kind of taking all the pieces of the puzzle and kind of laying them out on the floor, and then eventually you start to see some connections. So it's like this steady slogging, necessary evil, or necessary component, maybe not evil, necessary component of the natural sciences. But then there's this, flip to the applied sciences. But I'll tell you the thing about call it my research career, where science started to get really interesting, is when you start to, like, all these things that you explained mechanistically, right, about essentially protein purification, all of these mechanisms that are developed. I mean, it's like, I hear you talk about all these advanced processes, and it's kind of like, well, what would you think? Like Banton and Best, the folks that isolated insulin for the first time. It's like what you're doing is like on steroids, right? Amplified so much more and with such more precision. And of course, they had so many failed experiments because they did it once and they didn't really have the protocol down. But those protocols that you're building essentially, as you alluded to, lay the groundwork for all this applied science, but then also in the context that you were discovering all along also. And you're a guy, you have, I think, a love for is it jujitsu? I think that's what it was, right?
Brazilian jiu-jitsu, that is correct.
Yeah. So like when I talk to a guy who specializes in proteomics, and most people's understanding of a protein, you think, okay, the gene is expressed and it takes on this functional form, and here's this static molecule. It's like, no, these molecules are moving and living and breathing and interacting with other molecules. And you say they're these lipid components. Well, they're actually spreading through our membranes. These things are ionizing channels, are creating energy, right? So it's, mechanistically, you're getting to the point of, well, how is the genome expressed? And by the way, I think that's just one cell type you might be looking at. There's
Oh,
many types of cell types, many different organisms, and so it's like I'm sure we're still scratching the surface, but now we've got all these incredible mechanisms. We've got new ways to visualize, new ways to
yeah.
quantify with artificial intelligence, et cetera. And so, I'm interested to know what you're working on at this moment with respect to iMetabolic Biopharma, because I know you're working on a number of grants specifically. And first, of course, I want to acknowledge the tragedy of your wife passing.
Oh, thank you.
Yeah, I'm truly sorry. That is definitely a forcing mechanism. I mean, to a large extent, my mom passing with fueled a lot of my interest in trying to understand the relation between genetics, environment, gene expression, and how that all manifests into disease. And one of the things that I saw when we were doing all of this, call it gene surveying, is like somebody's going to have to do the science. Somebody's going to like the applied science to find out how this stuff all actually works, and which sounds like what you're doing. And so I'm interested to know like what's going on at iMetabolic Biopharma like this year, to the extent that you can share. I know it's a cardiovascular focus. Maybe we can talk a little bit about like pharmaceutical drug development in general, because I think up until this current moment, sure that this will change again with big data and AI and citizen research groups and data sets, et cetera. But the cost of creating a single drug is, I think, around $7 billion. And that's amortizing over all the failed drugs, right? So
Right.
maybe you could talk a little bit about what it actually means to take a therapeutic from idea to market, and where does iMetabolic Biopharma fit along that journey?
Yeah, I gotcha. And just to take a step back, there was a lot of stuff that you just kind of threw at me there, Jeremy. So I'm not quite sure exactly where to begin with. But I did want to make this, make one comment for our listeners. When I share the story about my first wife, and went through all of that, pain is something that is one, it's a great motivator. Number two, it creates resilience. And if you guys are familiar with a company called NVIDIA, the CEO, I'm blanking on his name, he gave a speech to a bunch of Stanford graduates, and he was talking about resilience and how that is the key differentiator between success and failure. And even though these kids were graduating from the MBA program from Stanford University, which is one of the pinnacle schools that you can get an MBA from here in the United States, if not the world, he felt that they were at a disadvantage because they didn't have resilience. And so he ended off with wishing that the most pain in the world that they can experience. Because if you can get through that, that will take you to the next level. And so, with these stories, with these personal strategies, and like, I can even go in, because you asked me the question earlier, Jeremy, like, why'd you go into science? Well, why did I go to graduate school? This was actually a plan B. I originally wanted to go to med school, but I was running into some headwinds, simply because like I grew up in Canada. I was an American citizen. I wasn't a resident of any state. And there's that preference for state residents. And I was a resident of nowhere, and nobody was willing to accept me unless I bought a house. But I'm like,
and out of state medical tuition rates.
Exactly. It's kind of it's kind of crazy with the way that worked. And so basically the reason why I went to graduate school was on my mom's deathbed. I promised her that this fall I will be pursuing a doctorate degree. And then, so again, please don't take anything that I'm saying. It's, oh, poor urban. No, it is what it is. And this is the cards that life gives you, and you got to play them. And you need to make the best out of them. And I'm a firm believer, whatever happens in life, good, bad, and different, there's a lesson to be learned, and, and I apply that into our research. It's like, okay, something failed. Well, define failure. It didn't meet your expectations regarding what your outcome was, but maybe your hypothesis was incorrect. Let's take a step backwards and look at the question we were answering, because maybe if we do a little tweak of the question, The answer's right
Hmm.
there. And that's a part of the reason why I've been so blessed and successful in my career, has been having that ability to roll with the punches, so to speak, being
Right.
able to adapt to it. It's like the data is the data. Well, I had this idea, didn't quite fit. Well, wait a second. Let's put a spin on
Mhm.
this. Now this works perfectly. And anytime I get a certain data set from anything, whether it be from consumer goods to, to even like in the biopharmaceutical space, it's like you have to ask the question. I understand where they're drawing these conclusions from, but are there other ways that you can get to that same endpoint without that explanation as to how they got there? And that's one of those things. It's like I want to see the data, and then I'm going to formulate my top 10 list as to how you got there. And does that
Right.
go and coincide with what people are promoting as well, this is the mechanism of success. So now talking about success, let's just jump into IMBP and what we're currently doing. You define the company as a biopharma company, and I do that too, just for simple or biotechnology company, just for people just understanding what you're doing. But in reality, we're actually is tech bio, not biotech,
Mm.
And
Right.
so,
Right.
again, it's 2024, things are changing. And even though biotechnology sounds really cool, it's some of the techniques that they're using they're older than I am. And like, I'm 50 years old.
Mm.
So
Yeah.
they're,
Yeah.
yeah, it's technology, but it's a lot of this stuff, it fundamentally hasn't changed. But from the tech bio standpoint, is let's take technology and let's use the technology to turn around and drive this the life science aspect of all of this. And that's at the forefront of what IMBP is doing. So we have our own proprietary platform, iPlatform, that's what our trademark name is. And it's an AI drug discovery platform in which we're able to computationally model and simulate the dynamics of our protein targets of interest in very complex interactions, and again, this goes back to what you were talking about. A lot of people just think it's a Lego block, and it's like, no, these things, they move, they're dynamic, they've got interactions this way in a certain state, and then they interact this way if there's phosphorylate, excuse me, I can't speak today. And then these
Mm.
things need to be accounted for, especially if you're trying to solve a particular disease pathway, because that's the way these things all begin. It's very, very subtle, because it has to be
Mhm.
very subtle so that the body doesn't, like in the case of cancer, it has to be very subtle, or else their body's going to turn around and key on, that's a foreign invader. We need to attack it and get rid of it. So
Mhm.
I view these problems that the body is undergoing is this, you've got these little kingdoms and stuff, and there's these constant attacks and repulsions, and what is making one succeed versus the other? Is it like a lack of resources? Is it, covert stuff in the case of oncology, and these tumors are able to go and just proliferate and invade healthy tissues and starve them of nutrients until it gets to the point where the organism, the patient is dying. And, it's like, what do we do at this point in time? Because now we can actually physically see it. The interesting thing with cardiovascular disease, and as I mentioned, we're cardiometabolic, cardiovascular, and neurodegenerative, more specifically Alzheimer's disease. There are these
Mhm.
metabolism components to all three of them. And this kind of explains why when you go and you look at things like obesity, which is now finally becoming something that is, front and center in terms of a healthcare perspective. It was always kind of like taboo. Like even at one point in time, they were unwilling to go and call patients obese or morbidly obese. They were calling them overnutrition. You're doing these patients a disservice when you're not
Mhm.
calling it out what it is, and trying to go and just give things warm and fuzzies, when in the reality it's trying to save a person's life. And so,
Mhm.
dealing with the lipid metabolism aspect, and a lot of lipid metabolism is kind of like trivialized out there. But in the case of
Mhm.
cardiovascular disease, oh, we got cholesterol lowering drugs. Yeah, you got more than you can shake a stick at. But why are 1 in 3 Americans obese? Why are 70% of Americans overweight and obese? Why are 1 in 4 Americans going to die of cardiovascular disease? Why are 50% of people who have heart attacks here in the United States have normal cholesterol levels? Have you really solved the problem? I don't know. It doesn't sound like it to me. And so, that leaves this landscape of, yes, things have been done this entire period of time. Has it helped out? Sure. But still, it's the number one killer, even though you have all of these treatment options. Something isn't panning out. So let's take a step back. Let's do some
Mm,
critical evaluation. And this is where things like iPlatform, as well as my experience that I've learned throughout the course of my career, have been able to go and develop some very interesting and innovative approaches to tackling these diseases. And so been able to develop some really strong relationships with key collaborators along the way in all of those different disease spaces, and they're very excited regarding the innovation that we're moving forward. But again, this is a long, hard fought process. And
mm,
you threw out some numbers. I think the last time I looked at it, just to go and get a single drug to market, and that's not accounting for making up for failures, other drugs, that need to be compensated for as a part of it. I think it's 2 million to go from initial discovery to, or excuse me, 2 billion with a B, to get it from beginning to the marketplace, and on average about a decade. And that's not sustainable, especially
mhm,
considering that people are dying, and because of the duration and these costs, it's leading to the development of therapeutics that are just, the average person can't afford them.
mm,
You don't hear a lot like regarding the cardiovascular space,
mm,
regarding cardiovascular therapeutics that are coming to market. Like everybody knows what a statin is, and maybe fish oils and stuff. Which that's a double edged sword, because the fish oils now they're showing cause AFib, and I was actually a victim of that one,
mm,
unknowingly, just taking fish oil
mm,
from Sam's Club to go and call them out. It's just these things that there are regular remedies, but we don't hear about like the innovation developing in that space. Sure, the new PCSK9 drugs, but now when those got released into the marketplace, that was a pay or cost of $16,000 per year. Most Americans
wow.
go and turn around and pay for that. The latest one is a drug called Evkeeza. Evkeeza. I'm butchering it. The names of some of these drugs are just ridiculous in terms
Mhm,
of being able to pronounce them. That drug price is $45,000
wow.
per month. Like, who
Wow.
can afford a $500,000 a year for a drug therapy? So again, we're ending up in this scenario because the way the system has perpetuated itself, in which the innovative drugs are just way too expensive, limiting who it can go to. Even if you get reimbursement from insurance systems, it's still a huge burden on the system as a whole. And this is one of my gripes when it comes to like the gene therapies. It's, these things are so expensive. Like the
Mm,
new, I think Bluebird had approval for sickle cell. Treatment cost is
mhm,
$4 million
wow.
for sickle cell, and they turn around and justify that is over the course of a patient's lifetime. It's like six. So the cost of the drug is 4 million per dose, but the lifetime of treatment based on averages is 6 million in treatment costs. So there's a net savings of 2 million. But the question that I have coming back is, show me your 30-year trial supporting that
Mhm,
there's no attenuation in the drug efficacy when it comes
mhm,
to
mhm,
all of this. Or are you going to have to do it
mhm,
again? Then you're at a net loss by 2 million on
mhm,
that. So it's just, it's one of those things that, there is the business aspect in which we're investing all of this money, and we have to get a product to market because we have to generate revenue. I totally understand that and I respect that. But because of these cost challenges, because things haven't kept up with technology, and this is where tech bio comes into things. Things can be done a lot better. You can streamline these processes, get them done faster,
right,
get them done cheaper, and in all honesty, make things better so that you have a more efficacious drug with lower side effects, in theory, to be able to
right,
help the patients
right.
out. So I hope I
Right.
answered your question there, Jeremy, because that was both a lot on both sides there to unpack.
Yeah, no, that was really helpful. I mean, I guess the question is, do you see a day with your technology where, I mean, it's probably the case. Well, since you're in business, it's probably the case that you already are reducing costs associated with drug development. So, and do you see a day of, like the cost to sequence a genome, reverse Moore's law, right? It gets, much smaller, exponentially smaller, fractionally halved every time based on the technology improves. Do you think we'll see like a reverse Moore's law with drug development?
The simple fact that we haven't seen it already,
Hmm.
I have to sit there and say no, because there's just so many steps when it comes to
Mhm.
getting these dinosaur companies. Like
Mhm.
these big pharma companies are absolutely massive. Like, so how do you turn around
Mhm.
and streamline them? You're caught between a rock and a hard place. Going and streamlining regulatory through government. Like, good luck with that one.
Mhm.
And these are the natural steps that you have to go through. That just even running some of these clinical trials, it's if you want to run, if you need, like for the FDA here in the United States, the Food and Drug Administration, if I need to have a power set that requires 5,000 participants in a drug trial, I need to recruit 10,000 people to account for attrition.
Yeah.
Does anybody have
Yeah.
any idea how expensive that is to go in
That's so
and
hard.
use?
Yeah. Yeah. Well, I
Yep.
know, one, let's say, collaborator I was talking to the other day around ALS, asymptomatic ALS recruits that they're looking for. The CRO is paying 25,000 per person to participate. Yeah.
Yeah.
Yeah. It's I mean, which in a, you know, let's say, framing it differently, say to individuals who are, thinking about, their own data and how your data, helps you make better decisions as it might relate to staying healthy. And hopefully that's the case that we're pursuing. But then, so you do have to take the treatment path. Well, you talked about the efficacy of the drug. Well, there could be adverse responses, as we know. But basically, as we start to, potentially get people to a place where they have all of their health records organized, maybe, and I'm speaking of a utopian society here, organized, they own it, they can even start to leverage it, and that becomes your side hustle. So, like, instead of, Uber driver as your side hustle, maybe selling your data as your side hustle. I mean, it's an interesting time and space, potentially, for that decentralization of data. And I'm starting to see like small companies pop up and providing that autonomy. But one of the things that I, struggle with is, great that these technologies exist to house the data, but it's garbage in, garbage out. And just the habit of, collecting and organizing your data, like, over your lifetime, that's a huge task. Sometimes I trouble to organize my calendar in a week. So I think there's definitely some logistics challenges, but I think the opportunity with more smart devices, maybe more collaborative centers of excellence, like I'm seeing that more in the rare disease community. Because, of course, like in rare disease, so few people have the disease, so there isn't much economic incentive to develop the cure, which, I think is a good segue into, your podcast, Decoding Health, because you're, a bit of a unicorn. You've got this, I'd say, call it a pure science background turned applied science business maven, and now you're, developing your voice as a thought leader in this space. So, tell me a little bit about the motivation for Decoding Health, and I mean, because you have no shortage of things to do. So,
Yes. Well, tell me about it. Tell me about it. Like,
mm,
again, I've got September 5th.
yeah,
I'm not sure what the delay is between, us recording and getting I've got literally 2 weeks to go in finishing up 2 SBIR proposals and getting them out. And
yeah, yeah,
a lot of people are like, well, you've never written a grant then, if you if you're
yeah.
thinking, if you're just trivializing that. It's a lot
Oh
of
yeah,
work, but
oh yeah.
here, nonetheless, I want to support you, Jeremy,
Mhm.
and so I'm on the show right now and
Mhm.
sacrificing a precious
Mhm.
day to be able to go in and do these things with the podcast. But, just regarding the podcast, when I founded IMBP, because you got to remember, I left Thermo Fisher because I had a job that was killing me. If you saw pictures of the way I looked then to the way I look now, you wouldn't recognize. Like I had a beard because I was unhappy with the way my face looked because I had a double chin, And then now, like well, I'm not competing right now, but like when I get into shape and competing, like I've got a full six pack. I compete at 168 and BJJ, like at the international level, and it's like, okay, this is my commitment to me. But even though I own a drug company, I'm the founder and CEO, I have a promise to myself, a promise to my family that I'm never going to be a customer of it. That means I have to own my own health. And just when it comes out there, in order to go and drive success as a society, because again, remember the numbers here in the United States, we pride ourselves on being number one. Well, to be number one in obesity is not something that one should be proud of. But yet here we sit, number one in obesity. What do we have to do to drive societal change? Because one, it's better for our people. Our people will be more productive. That adds to GDP. You end up with a stronger country. It takes the financial burden off of the healthcare system, off of the social healthcare system from, the government standpoint. Lower taxes, people have more money, they feel rich, they spend more. It's this improved cycle, but it all begins with the individual owning themselves, getting themselves in order. And this is actually how you and I got introduced, is through the world, through Kathleen Ferguson. Thank you, Kathleen, for getting us connected, because there is this huge mismatch when it comes to the biopharma world where I come from and the fitness space. And most people view them as this adversarial aspect, when in reality, there is a huge mesh there, but both sides need to sit down and start talking. And, I've got my views, personal views and thoughts. And I really think if this is done correctly, you can drive fundamental change to go and make everything work better in a positive fashion. And again, here in the United States of America, let's be number one. Yes, let's be at the forefront of this, because it's not just the US that's suffering from this. Canada's got an obesity issue. Europe has an obesity issue. It's in the Middle East right now. The UAE is well on their way to trying to go take us out from number one. And there's such overlap in these different diseases, because they cluster together. But they are well on their way of a huge rate of obesity. And it's really easy for people to go and say obesity, type 2 diabetes, cardiovascular disease. Okay, fine. They're expecting by 2030 a 600% growth in Alzheimer's disease.
Wow.
Because people don't go and make that connection regarding fat metabolism and the needs of the brain. And it's just like, it makes all the sense in the world if I've got the luxury of having decades worth of studying this stuff, but it makes all the sense in the world to me. And again, going in and understanding this stuff, like what the driver for the Alzheimer's program within IMBP actually started through what happened in 2020. I'm not going to go and say the name of what it was, because your podcast will get flagged and end up in purgatory. But what we went through all of that, in the whole systemic inflammation that resulted from that, and they've got that long version of it resulting in brain fog and confusion on that. That really made me do a deep literature dive into all of this. And there's this connectivity over into Alzheimer's disease. And it's just like, that's kind of funky. Where does this go? And then, oh, wow, there's this huge lipid metabolism component. And when you have this infection and your lipid profiles go out of whack, and if you sit down and look at the way those things go out of whack, not just from a total number of a particular lipid, composition, but look at the sub-factors when those things get dissected out. It's, again, you don't have to be Einstein to, in my opinion, to figure this stuff out. You just need to be paying attention.
So, yeah, I mean, It sounds to me the Decoding Health podcast it's like the high touch complement to your high tech work. So it sounds to me like you've been educated. You said you have multiple decades of education in here, and now you're getting the word out, and you're connecting collaborators. So I know we only have a limited time today, so I wanted to, get your information. So listeners who have heard this and are inspired by this, what is the best way for them to find you? So I know there's your podcast, Decoding Health. We've got your company website probably as well that we'll definitely put in the show notes. So besides iMetabolic, Biopharma, your podcast, what's the best way for people to find you? Is it LinkedIn? Are you on social? What's the best way?
LinkedIn is probably the easiest. It's Urban A. Kiernan, PhD. I'm the only Urban Kiernan on there, so I'm really easy to find. Again, too, if you're looking for the Decoding Health podcast, go to YouTube, just search Urban Kiernan. There's also links there to like Spotify and everything else on that. You can find me on social media. My Instagram is actually public. That's under Lightbearer. That's my little pseudonym that I go by. I like to illuminate things. We actually talk about that on the podcast. And again, too, it's just I'm just doing my best to assemble voices from all of these different segments. Let's come in, let's get these messages out there, and be able to go and again drive something to make this. Someday, I'm going to be gone and dead, and I would love to be able to make this world just a little bit better when I leave than when I came into it. And, there's all of this negative stuff that's going on right now, but, there's some huge opportunities to go and bring some really fact-based science to the limelight that can really, really help improve people's lives. And if this was going to be my last podcast,
you beat me to the punch. You beat me to the punch.
If it's one thing that I could tell people to do is to cut sugar out of your diet, please. I'm begging you, it's cut sugar out of your diet. I'm not saying go full keto or anything like that. Just reduce your sugar consumption. That messes everything up. You don't have to be type 2 diabetic for this to be an issue. It messes up your lipids, and I'm sure you're going to wind up getting, flack from people. It's like, oh, your brain needs sugar to go into. Okay, it does and it doesn't. Actually, your brain prefers ketones, but let's not go and go down that road. But just cut the refined sugar out of your diet. That is a huge step forward for everybody.
Well, that's a simple wrap. Let's see if we can have some effect there. The one last question is, who would you nominate to be on the DNA Things podcast?
To be honest with you, I've got too many names to. Yes. So to be honest with you, one of my guests and advisors to IMBP, Dr. Malu Tanzi from University of Florida, moving to Indiana University, Alzheimer's, Parkinson's expert. She is absolutely superstar, wonderful. Also at University of Indiana, Dr. Paul Torito, ex Eli Lilly. So he's made the bridge from industry over into academia. Again, cutting edge whiz when it comes to all of this stuff. If you could do it, I know it's not really his thing. Dr. Ronald Kraus from University of California, San Francisco School of Medicine, also advisor to iMetabolic Biopharma, is the foremost expert when it comes to dyslipidemia and abnormal lipoprotein particles. Absolute wizard. And I love the guy because his whole body of work over his entire career challenges the status quo. And if you go so the status quo is one thing, and there's a million different reasons why that status quo is out there and that narrative is pushed. Some of it's right, some of it's wrong. When you go and you speak to him and you listen about this, he's more right than wrong. And so it's just like this, again, driving fundamental change. You just can't keep on going with the same narrative. You got to mix things up. So, it's those are those are my three names that I would throw out there immediately.
Oh, wonderful. I'll definitely reach out. Well, listen, Dr. Urban Kiernan on the DNA of Things, thank you so much for joining. Thank you for being so generous with your time, and good luck on your grant applications this fall.
Thank you. Really, really appreciate it.
All right. Thank you. As we close out today's fascinating conversation with Dr. Urban Kiernan, we've traversed the future of medicine, the intricacies of proteomics, and the untapped potential of lipid metabolism in disease treatment. What stands out is how science, technology, and personal resilience converge to push boundaries. Dr. Kiernan's story is not just one of scientific discovery, but of perseverance, personal transformation, and a mission to make healthcare more accessible and affordable. From understanding how proteins are the key to unlocking many diseases to rethinking how drugs are developed, today's episode reminds us that health is a lifelong journey, one that we can all take charge of, starting with small changes. Now, if you found this episode valuable, you value the conversation, please share it with your friends. We are building an organic community of conscious creators. Together, we can continue spreading the love of knowledge and make a lasting impact. So stay curious, stay healthy, and as always, thanks for listening to the DNA of Things.